Hi, my name is Felipe da Costa, and here I will present a study developed by me, Maria de Fátima Arruda, Karina Ribeiro, and Daniel Pessoa at the Federal University of Rio Grande do Norte, Brazil, entitled Influence of Color and Brightness on Ontogenetic Shelter Preference by Prawns, Macrobrachium rosenbergii. Macrobrachium rosenbergii, the animal that can be seen here, is a commercially important species of prawn cultivated in several countries around the world. A characteristic of this species that makes its cultivation difficult is its great aggressiveness. The provision of shelter, like here, can reduce the frequency of agonistic interactions and improve the culture. Thus, it's important to study the preference of the prawns for different shelter characteristics. And we wonder what the preference of prawns is for different colored shelters. We based our prediction on the life cycle of these animals. Juvenile and adult prawns live in freshwater environments. Original females migrate to estuaries where eggs hatch, giving rise to larvae. These undergo metamorphosis, giving rise to post-larvae that migrates to the freshwater environments, giving rise to juveniles and later to adults. So, we predicted that juveniles and adults would prefer longer wavelength colors, such as yellow and orange, which are characteristic wavelengths of freshwater environments. In nature, this preference could help the prawns remain in freshwater environments. We brought juvenile prawns from the Agricultural School of Jundiaí to the Sensory Ecology Laboratory of the Federal University of Rio Grande do Norte, Brazil, and kept the prawns at an artificial light cycle of 12 hours light and 12 hours dark, with the light phase starting at 6 a.m. We offered commercial feed twice a day, early in the morning and late in the afternoon. We tested 39 juveniles about 2 months after metamorphosis, and we retested 24 of these animals when they were adults about 7 months after metamorphosis. We tested the animals individually in aquaria containing 2 rows of 5 shelters. There was a blue shelter, a green shelter, a yellow shelter, an orange shelter, and 6 gray shelters with different brightnesses. We measured the reflectance curve of the shelters with a spectrophotometer, as we can see in the figure. The abscissa shows wavelength and the ordinate shows reflection percentage of each shelter. Also, we measured the illuminant of our laboratory. Illumination was provided by 2 fluorescent lamps. In this figure again, abscissa shows wavelength, while ordinate shows photon flux. Next, we modeled the vision of the prawns. For this, we took into account the illuminant of the laboratory, the reflectance of the shelters, and the absorption peak of the photoreceptors of the closest species for which these data are available. This species is Palaemonetes peludosus, which has a photoreceptor with an absorption peak at 380 nanometers and another with an absorption peak at 555 nanometers. On test days, we transferred the prawn to the experimental aquarium at 8 a.m. From 9 a.m., every half hour we recorded whether the prawn was in or on one of the shelters, like here. At 5:30 p.m., we made the last record and removed the prawn from the experimental aquarium. The position of the shelters for each animal was random. We used Biostat version 5.3 to perform the statistical tests. We performed 10 chi-square tests to verify if each shelter was occupied at the expected frequency, which was 10%. We employed the Bonferroni correction. Thus, the significance level was 0.005. This graph shows the results of the visual modeling. Abscissa shows achromatic signal. The higher the achromatic signal, the brighter the shelter in the vision of the prawn. Ordinate shows chromatic signal. The higher chromatic signal the more the short wavelength photoreceptor is stimulated when compared to the long wavelength photoreceptor. We can see that the green, the orange, the yellow, and the blue shelters have very similar brightnesses in the vision of the prawn, since all they have very similar achromatic signals. The green, the orange, and the yellow shelters have very similar chromatic signals, and they have very different chromatic signal when compared to the blue shelter. This figure shows shelter occupancy by juveniles and adults. Abscissa shows the color of the shelters, and ordinate shows shelter occupancy. Black bars represent shelters occupied more frequently than expected. Gray bars represent shelters occupied at expected frequencies. And the white bar represents a shelter occupied less frequently than expected. We can see that juveniles occupied the blue shelter more frequently than expected and occupied all other shelters at expected frequencies. On the other hand, adults occupied the two darkest gray shelters more frequently than expected, occupied the brightest gray shelter less frequently than expected, and occupied all other shelters at expected frequencies. Our work was the first to model the vision of prawns. Our prediction of preference for longer wavelength colors was not corroborated, since juveniles preferred the blue shelter and adults preferred the darkest gray shelters. The preference of juveniles was based on chromaticity, since they preferred the blue shelter over the green shelter, the yellow shelter, and the orange shelter, which were very similar to the blue shelter in brightness and very different chromatically in the vision of the prawns. On the other hand, the preference of adults was based on brightness, since they preferred the darkest gray shelters and avoided the brightest gray shelter. We recommend providing blue shelter for juveniles and dark shelters for adults in order to improve animal welfare and productivity. Also, we encourage the development of new studies in the area to improve our understanding of the preference of these animals for different shelters along the ontogenetic development in order to improve the culture of prawns. If you have any question, you can contact me by this email. Thank you very much.